familiar with in Mexico here. This is called a cool temperature turf grass. This is Agrasta stolonifera. Common name is creeping bentgrass. In Canada and the northern United States, it's primarily used for putting greens. So here is a golf course. And if you're familiar with golf courses, then you have your tees, etc. But you have here, which is the key element, you're trying to get the ball in the hole here. And so you do that final step where everybody's very quiet and they do the putting into the hole there. Well, that grass is creeping bent grass because you keep it extremely short. You basically cut it every week. And there's a number of diseases on this which can cause dead spots in the grass. And if there's a dead spot in the grass, your ball rolls and then rolls off sideways when it hits the dead piece of grass. So people like Tiger Woods, etc., get all upset, right? Because they're paid millions of dollars and suddenly this disease makes their ball go the wrong direction. As a result, there's essentially zero tolerance for disease in these putting greens. And the result is that it has the heaviest use of fungicides in the United States and Canada compared to any other crop. Basically, if you're a turf grass manager and you get dead spots on your putting greens, you're fired. Okay, so it's a pretty serious thing. And also you have a lot of stress on it because you essentially have to mow this every week because you can only have the grass about 5-8 millimeters tall maximum. Okay, so we worked a little bit on one of the diseases, snow mold, Microdochium nivalis, in terms of diseases. And we work mostly the common SAR activator, the BTH, and then the ISR activators, the butanediol, BD, and the civitas. And there's previous evidence that there is variation among turfgrass varieties in terms of their effectiveness or responsiveness to these compounds. So what we looked at was then comparing cultivars of creeping bentgrass. Can we see that different cultivars will differ in terms of their induced resistance? And then ultimately see if this relates to changes in defense gene expression, because that has been shown in tomatoes with Pseudomonas syringae. And so we call it responsiveness, which basically is how much disease do we have when we don't do an activator compared to how much disease we have when we do an activator. So we set up in the laboratory, this is little vials of turf grass growing in the laboratory here, small plants, and then we inoculate with this pathogen called brown ring spot of turf grass, and here we can see, here's a putting green, and you can see these little yellow areas where the grass is dying, and it forms on the edges of it, so it gives a little bit of ring appearance. Once again, this is a zero tolerance disease for putting greens, and there it is in culture. So what we did is we grew some plants up, we treated them or had them with water. A week later, we inoculated with the fungus, and then 2 weeks later, we determined the amount of symptoms. And basically divided up into those at strong response or weak response. So here's an example, the cultivar Brighton. You can see compared to the water control here, lots of disease. With BTH, very low levels of disease, compared to another cultivar called SRX1, where the BTH is not working at all. So you can see that this has a major difference, and this, of course, has a practical implication, because if you're buying this product and you're growing Brighton, you're going to be very happy with the product. If you're growing SRX1, you're going to be very unhappy with the product there. And then we did the same thing for the butanediol, and once again we could find cultivars that respond well to the butanediol and ones that don't, and these are different cultivars than for the BTH. So out of this, our screening, we picked 2 of each, 2 that strong response, 2 that weak response. And you can see it differs, right? Because PEN-A4 responds very well to BTH and responds very poor to butanediol. All right, then we wanted to look to see if there's some changes in gene expression. Now, for the BTH, the problem is we didn't have any target genes, so we did some Illumina sequencing. This is an older version of the machine at the time. And we got a whole bunch of reads and looked through that. So this is when we did the RNA, when we treated our plants there. And we had about 2,000 DEGs for that responsive, right? A lot of them, the problem with monocots, we don't have much of a description, but we found 17 potential defense genes, and out of this, we chose 3 then as our targets for comparing responsive and non-responsive lines. And then we used RT-PCR. Ubiquitin was our control. And here we can see some of the gene expression here. And then we looked at those genes and tested them in the weak and strong responders. And here you can see some of the responses here. Green is with the BTH, blue is with just the water. And if you look at it, you really can't see much of anything obvious difference between the strong responders and the weak responders. Nothing consistent between the 2 varieties. Aspartic acid, the ASP2 gene, once again, nothing really jumps out at you that one responds with the expression, one doesn't. And with the HIR, once again, nothing you might say, well, this variety has a very sharp early peak, but here this weak one has a very sharp early peak as well. So this is just 3 of those chosen genes there. So in the end, it really is hard to say that there was anything obvious in terms of gene expression of the 3 genes we chose that could explain the responsiveness. Now for ISR, with the butanediol, we had previously published, had looked at some genes in creeping bentgrass that showed a response to an ISR. So we looked at the high and weak responders. Once again, here we have one of the genes, very similar, nothing really jumps out different between the strong and weak responders. A second gene, once again, nothing really jumps out. This is a little bit where expression is not above the control, but here it's a little bit above the control. So there's nothing really that obvious here. Another gene, once again, quite similar. Here we have strong responders, which really are showing nothing above the control. And so on the positive side of this study is we're able to show that cultivars of A. stolonifera both respond differently to BTH and butanediol, so SAR and ISR. There's no consistency, so it's not that something responds well to all the different agents. So this, of course, has a practical implication. If you know what cultivar is on your putting green, you would want to know whether the product you buy, like butanediol or BTH, actually works. However, we didn't find any relationship, unlike in the tomato work with the PR genes, similar PR genes to what we found in terms of the responsiveness here. That doesn't mean that there aren't genes that are involved, but we didn't find them in the types that we had examined there. Okay, so that's the first part, looking at the creeping bentgrass. Now, another thing that we were interested in is something called chlorophyll fluorescence imaging. And so over the years, actually since I was a graduate student, one of the things on and off with different projects I've worked on is just different ways to quantify diseases in terms of their severity. And this is another method, chlorophyll fluorescence. You can measure chlorophyll fluorescence just numerically with little meters, but this actually has a camera and so creates a false color image that you can see entire plants or entire leaves there. So here's some previous work we had done. And in this case, we're looking at various diseases. And what you can see is you got lesions on here. This is an anthracnose of a monocot. And you would estimate, okay, there's this amount of disease, this amount of disease. And typically, people estimate either as a percentage or they take ranges and they make a category like 1 to 5, etc. In this case, what we're able to do is just take a regular digital camera, put them into a standard computer program, convert the different colors into false images. So this is the same thing as here. And then use a program, image J, to quantify the relative amounts of the different colors in the false image. And so this is a way to get something that's less subjective, right? You would probably say, well, this one is about 25%, this one is about 50%. Although the 1%, most people would overestimate that, because once you see a small amount of disease, you start to say, wow, that would be 5% or whatever. Quantifying it gives you something much more reproducible. So just as a review, one way to quantify pathogens, for example, is to label them, such as with GFP, and then you can extract the GFP and actually quantify it in a fluorometer. We did some work on that. This is some previous work. This is cholototricum. And so we were actually able to show with the GFP labeled one, rather than just relying on image analysis, you actually grind up the leaf, extract out the GFP, put it in a fluorometer, and that can measure good amounts there, going from the initial infection, finally up here to spore production. You can see some conidia, and then you can see some CT here, which is typical of your cholototricum diseases. So visual ratings, subjective, that's why people usually use ranges rather than specific numbers. The computer image analysis, like I showed you there, is much more accurate, but of course then you need to have symptoms having developed. It doesn't detect pre-symptomatic tissue. And the fluorescence tagging, like GFP or whatever, is fine, but you first have to work at tagging the organism to detect that, but that works fine. So that's a little bit time consuming if you're working with a wide range of pathogens, and is obviously not going to work if you're doing something in the field with natural infections, because no one's going to let you go out and start spraying GFP labeled fungi around to inoculate out in the field there. Well, not legally at least. Okay, so that's a bit of an introduction here. So one of the things then as an alternative is chlorophyll fluorescence. So when you have light hitting leaves, we all understand that you get absorbed by the photosystem, we get our photochemistry, and that starts to generate energy for our plant there. However, less appreciated is there's actually two other ways that that energy can be used. One is it's simply lost as heat, and the second is that it's lost as chlorophyll fluorescence. So very often the amount of light is in excess to the amount of ability of the plant to turn that into phytochemistry. And once again, if you're familiar with plants, this is fairly obvious, but basically you have this, it's all a membrane-bound system. You have the light hitting those membranes in the chloroplast. You have your light harvesting machinery. You move your electrons through, eventually getting to production of your ATP. And that's what you want, photochemistry, right? But it's just often it just is not able to absorb all of that energy and turn it in effectively in the photochemistry. Heat is generated and fluorescence is generated. Now, the photochemistry really has a maximum, so as you keep increasing the amount of light, what you are doing is not increasing the amount of photochemistry, just increasing the amount of heat and fluorescence that's coming up there. So you can't really say, well, we'll increase crop yields by just increasing the amount of light. Plants are adapted, shade-adapted plants, sun-adapted plants, etc. So the more light, not necessarily more photochemistry, just more heat and more fluorescence. Now, that makes it possible then to measure this fluorescence, because we know the wavelengths, especially when we control the wavelengths here. That fluorescence has been established to be affected by leaf pathogens, well, even root pathogens, and it is pre-symptomatic when you see the changes, so you don't need to have the visible symptoms. Once again, you can measure this non-subjectively. Well, you can't measure it subjectively. You can't use your eyes to see the fluorescence. And with the equipment that's been developed for it, relatively quick to do, and these equipments are designed to work with a variety of plants. There's field versions as well as laboratory versions. Now, there's a lot of different parameters and ways of measuring chlorophyll fluorescence. I won't get into a lot of details, but FO, which is the minimum fluorescence, you put the plants in the dark, also called dark adapted. FM, the maximum fluorescence. Once again, it's dark adapted, but you give an intense light, which basically overloads that phytochemistry as much as possible. And FM, where you don't do a dark adapted leaf there. So those are all common measurements. More common is calculated measurements, which you take FV, which you take the maximum and minimum, or FV over FM, and then this one is often used as considered the maximal efficiency for which photosystem 2 is able to use light and then get into the electron transport. Here's a plant that you see around quite a lot because of virus-induced gene silencing, Nicotiana benthamiana, native to Australia, originally studied primarily for virus diseases, used heavily for VIGs as well as for transient gene expression. It does contain nicotine, so it was actually used by the Aboriginals prior to Europeans, but it really isn't used commercially anymore. And the pathogen we're talking about here is called Genotypic orbiculaire. It's a fungus. It is hemibiotrophic, which means it has several days where it grows by feeding on living cells. There's no visible symptoms. After several days, you get what you see here, which is a water-soaked lesion. As it turns necrotrophic, that will eventually die and form masses of spores in the center. And the disease is called anthracnose blight. Probably in Mexico, people are most familiar with anthracnose of mango, which is you have a nice yellow mango and these dark black spots in it. That's anthracnose there. Anthracite, coal, that's how it gets its name for the black part in there. There's nothing wrong with eating it. Calototricum doesn't produce mycotoxins, but obviously people don't go looking around for mangoes with black spots in them to eat. And so this is a good system because we know there's pre-symptomatic tissue in the biotrophic phase, which you can't see anything visible happening in the plant. So now we're looking at this disease as well as using the butanediol to trigger ISR. And we want to see how we can quantify this. So what we did is we took the images. We have a little machine there. We measured FM, FO, FM prime. And one of the problems if you use the direct measurements is it's very sensitive to the setup of the machine. The leaves have to be exactly in the right orientation and the same distance. And one of the problems here is you can see some lesions, but you can see they're marked as blue, but the healthy region is various heterogeneous, and you can see some green areas right around the lesions, which are affected tissue. Another one is you can calculate a measurement NPQ. Once again, we can see this is the real image, this is the false image, false color image, the black spots, but once again, rather heterogeneous, or even though this is a calculated measurement. FV over FM, once again, here's our lesions. We can see them as the false blue color. Yeah, it's more consistent. We have that green area, which seems to be just surrounded our lesions. And it can't see in here, but some of these areas which are just green were observed then in a day or two later to develop into symptoms. So we can see that a lesion first develops like this as a small green spot, then like this with a green spot with a light blue area, and then like this where you actually finally see symptoms with a dark blue area with the green around it. And because it is a ratio, this equipment is not so sensitive to distance, leaf orientation, etc. So based on this, healthy tissue was classified as red and orange. The tissue adjacent or the affected tissue is our green to yellow, and necrotic tissue is a blue to black false color image there. And so here we can see a false color image of some lesions which are blue, and you can see the surrounding the affected tissue. And then we could take this in a program and work with that in terms of quantifying the amount of blue tissue, quantifying the amount of yellow, false color, and then the total area of the leaf to get the amount of total images. So we went with this FVFM imaging system. So here we can see then examples of a number of leaves measuring those three areas. And what you can see here is that at 48 hours, everything is green, orange, or yellow, so nothing seems to be happening yet. At 72 hours, which is when we know that the biotrophic phase is occurring because we've done microscopy, we can now start to see these more stronger yellow spots in there. And by 85 hours or so, 86 hours, we now have necrotic lesions in there, and you can see a lot of affected tissue where necrotic lesions are going to start up again. So you can see that over time, we can see that it takes a while to see necrotic tissue, but we can start to pick up some differences in affected tissue well before that. And finally, we have at 96 hours, lots of necrotic tissue with lots of affected tissue around that. So this initial phase, up to 72 hours, is essentially the biotrophic phase, essentially very little or no necrosis. Then we have a transition phase, and we eventually have the necrotrophic phase of infection. And one of the nice things that we've established before is that the inoculum relates to the number of lesions there. And so by doing this quantification with the fluorescence, we can show a pretty good regression ultimately between the amount of necrotic area measured with the FVFM versus the number of lesions per area of leaf. Okay, so establishing that now, can we use that as a way to assess one of these activators, in this case, the butanediol, which activates ISR? So here what we can see is that if we look at the amount of tissue, we're just looking at it, you can see that with butanediol, eventually we're going to get a drop-off in healthy tissue, but it's delayed compared to the disease. Then the amount of affected tissue is delayed, once again, in terms of the amount of disease. And the necrotic tissue increases faster with the water, as you would expect. One of the interesting things, so it shows that we can quantify, one of the interesting things that we observe between the leaves with the false color images is shown here, that even when you eventually get disease on the butane diol plant, remember, just push back in time, if you compare the lesions, we quite consistently saw that the zone of false color for the affected disease was always much wider for the water control compared to the butane diol. In fact, with the butane diol, very often we saw almost no amount of affected tissue surrounding the lesions. And just remember, these lesions expand over time, so this tissue in a day or so will become necrotic, and then the affected tissue will keep expanding out from there. And so what we did was image analysis, cross-sectioning through lesions and measuring the amount of FVFM. So if we take a healthy leaf, that's the red here, you can see it fluctuates a little bit, but pretty consistent false color across. If we do the same thing, a transect for our water treated, what you can see is that, yes, of course, and when it's dead, there's no FVFM, but you can see that there's a gradual drop off here and then a very quick drop off. If we see the BD ones, we can see that that gradual drop off the affected tissue on the side is much narrower. And of course, when the tissue is dead, the tissue is dead. So this is just an example, but we did this a number of times, have the statistics on it to show that, in fact, using this, it's not just that the lesions are delayed with the ISR, it's that the amount of affected tissue is delayed, which of course is good because once the tissue is affected, they're going to be much less effective in photochemistry and phytochemistry, so they're not going to be producing as much sugar in there. So this shows that you can use a method like this to quantify the disease even before symptoms appear, and be able to detect something you could never detect visually, which is pre-symptomatic tissue differences between a treatment and your control. So chlorophyll fluorescence works well. We can distinguish healthy, affected, and dead tissue using the FVFM ratio. We can use this to quantify it. We can quantify things that are occurring even before symptoms are developing. And then using BT as our ISR activator, we can quantify the amount of dead and affected tissue and even show that BD not only delays the disease, but also delays the development of affected tissue and maintains tissue healthier for a longer period of time. Okay, this was supported by the Natural Sciences and Engineering Research Council of Canada. The student who worked on this was Jonathan Tung, who was a master's student, and he was advised by myself and Tom Shung, who is another plant pathologist at the University of Guelph. Okay, thank you, and that's everything. Okay, are there any questions?
Hi, Paul. So when you do the BD treatment, do you have like a dose response? I mean, if you increase the amount, do you have a better resistance, or there is like a threshold?
Yeah, there's basically a threshold. The BD is essentially gaseous. Okay. So you kind of put it on the plant and then have to seal it up so that the BD gas is there. At least with the BD, it's not as bad as the SAR activators, because the SAR activators, like BTH, have a very narrow range, and you just move a little bit above the range and you get toxicity. You move a little bit below the range and you get no effect. The BD has a wider range, but it's not a case that you can just keep applying more BD. So one of the problems in SAR and ISR is it's not like a regular fungicide where you apply more and you get more. It's like it has a narrow range. You drop a bit too low, nothing happens. You go a bit high, and you get toxicity.
So BD is chemically synthesized or can be produced?
Yeah, so BD is chemically synthesized. I think it's actually a fermentation product. And so what it's used for commercially is in the cosmetics industry.
Right.
And so that's why you can buy it commercially there, but it's, yeah.
So my second question is regarding the fluorescence of the chlorophyll. Do you think that there are some other fluorescent compounds, for instance, when you have the early stages of the infection, probably some phenolic compounds that are being accumulated in the region that could be used also for detecting those places that is going to be infected?
Yeah, so I mean, there is other work of people that have done things like, wire-run fluorescence in broad beans has been used, and you just use regular fluorescence microscopy because wire-run with a phenolic ring on it actually has a good excitation and detection wavelength. In this case, it's not other things because you really control, you have dark adapted, you have particular wavelengths for exposure and wavelengths for detection there.
Okay, thank you.
Another question? Here? No? Well, I have a question.
Hi, thank you very much for your presentation. I have a question about the image analysis. Did you use any database for this? I mean, not database, but like different image base that you have for the symptoms, or it was based on your own pictures of symptoms?
Yeah, so it's based on our own pictures of symptoms. We primarily use image J, which is a common, common publicly available image system there. So the main thing was just, so even those first ones with the symptoms is just a standard digital camera. So you don't need anything fancy. The later one with the chlorophyll fluorescence, you need a PAM meter. Certain companies like Walls in Germany sell those, so that has to be specific. But primarily, we have a program which you set your various cutoffs for where you define your false colors. And so you're just really comparing your original image, for instance, to the different false colors. Once you set those parameters, then you plunk it into image J. So that's the nice thing with that one with the digital camera. You could use anything. You could use your cell phone camera. Image J is publicly available. It's just for each individual gene. You have to do your settings there. But for the most part, it's not much variable because most fungal and bacterial diseases, they create brown lesions. So the colors are pretty consistent and yellow halos around them.
Did you use this technique for the graphs that you showed at the beginning of your talk for the golf grasses, or is it very? I mean, it's a very different plant from Nicotiana, but I don't know if it's useful for those small plants or not.
Yeah, so we did a little bit of work with that with the small plants. So it's not really a problem to use, although they are very tiny leaves. It's better to use the ones out in the field, which are a bit wider. But yeah, you can use them for basically any size leaf. It's really the PAM meter is really a little chamber, and so you can adjust the height in the chamber. You even have microscopes that have the PAM system attached to them, so you can even look at individual cells with the PAM system there. So we didn't do that because the PAM machine that we have, it's not so great for the smaller ones, but if we had a different PAM machine, they can do much more magnification of the images. But you can see that cross-sectioning, we actually were able to do just cross-sectioning of just millimeter distances in a leaf there.
Okay, we have one question from Esperanza Martinez. Have you tried chitosan or mixtures of elicitors?
So we haven't really done too much with other types of ISR activators. We were, primarily just interested in the SAR and the ISR themselves compared to the other materials. We did try a bit with the chitosan. One of the problems with the chitosan, especially for some of the naturally derived compounds, is like the chitosan, the acetylated form of chitosan works well. The non-acetylated form of chitosan doesn't work well. So our initial work, we had gotten a supplier in Quebec who extracted it from shellfish, and they were using it once again for the cosmetics industry, and it worked really well, and so we were very happy. That was our initial work on that. Then, for whatever reason, that company went out of business, and then we started to get some from other companies like Alpha Azar and Sigma, and then we had it didn't work anywhere near as well as what we had from that company. Some of them worked a bit, even some of them, even though it's Sigma, you buy different batches. One batch worked well, one batch doesn't work well. So we eventually gave up on that because that was just really too much of a problem. Even the butanediol, we've had problems with that, with a batch from Sigma that works really well and a batch from Sigma that doesn't work that well. So that really is a problem unless you prepare it yourself, because we contacted both Alpha Azar and Sigma about this. Basically, they just kind of shrugged and said, well, we buy it from other suppliers. They tell us they make it the same way. It should work the same way. I don't know if it's not working the same way. I don't know what the problem is, So that gets to be a problem with some of those ones if you don't produce it yourself. So it's just unfortunate we had one really good supplier. It worked just really terrifically, and we've never been able to get a chitosan purchase since then that works anywhere near as well what that company is. So things like the BTH, Syngenta makes it, it's purely chemical produced. The butanediol, much more consistent because it's produced for the industry. There's a big market for it. That's the main difference there.
What about directly the salicylic acid?
Yeah, most people don't use salicylic acid. Most people use BTH. Our experience, the same as in other people, is the Goldilocks range for salicylic acid is really narrow. And so you just touch a little bit above that, you're going to get lots of tissue necrosis. A little bit below that, you're not going to get any effect. So even though we complain that the range of effectiveness for BTH is narrow, it's not as narrow as what the salicylic acid is. So relatively few people use salicylic acid in these studies just because it's so tricky to get the range properly. And one of the problems with BTH, and one reason why Syngenta's really stopped working on it, is what they found is in the field, the problem is even in the laboratory or the greenhouse, you can get the range, but in the field, if you have plants that are undergoing stress, like water stress or insect damage or whatever, that shifts the optimal concentration. And so what they had was they got sued by certain growers because they applied it at the recommended concentration, but they had all this water stress at the same time. It caused massive necrosis in their crop. And of course, then Syngenta's on the hook for that, for the losses of that. So that's made it very difficult. So even though, say, 20 years ago, there was a lot of talk about these things replacing conventional fungicides, it's mostly at this point more for scientific interest. It's just too difficult on a commercial level to get that right concentration every time. And you can see with the turf grass, right? You're one golf course owner, you're really happy about it because you have a variety of grass that responds. You're another one, maybe just next door, you complain that it doesn't work at all, but it could be because you have a different variety of creeping bentgrass on your golf course. That's really different from a conventional fungicide where you don't have to worry about that sort of thing, that you don't have to say, well, this will work if you have this variety or that variety. So those are all become major issues in a practical sense for that. So lots of people keep studying Trichoderma, Pseudomonas, Bacillus for ISR, but it really gets hard to then translate that into a commercial product.
Any other question? I have one. Actually, I was intrigued with these grass phenotypes, the different varieties. Do you know if there is any, or if somebody studied the genetic basis of the difference? Because you observe and you focus only to check the expression of different defense-related genes individually in each of the varieties.
Right.
But what about the genomic changes in each variety between the susceptible and resistant? Since you have so many different ones, have you ever tried that?
So we didn't do something like that, but other people have done that. In terms of the genetic background, I mean, the problem is that people don't really reveal what the progenitors are for these turf grasses. But we did a similar study in tomato, and what was interesting was even though we couldn't get all the background information there for tomato, what we found was two companies that produce seed, commercial seed there. The products of one company, the varieties of one company, we tested several of them, were all highly responsive. And another company's varieties were all non-responsive. And so from the amount of details we can get, they have some similar backgrounds, but some differences in their background. So it seemed like that one company that where really responded well, they had brought in a wild species in their crosses of tomato, whereas the other company had not. And so we expect that this wild South American tomato may have been the reason for that responsiveness. So it's something that we've talked about to people from the industry, is that if you really want this system to work, you have to view it as an entire system, right? So yes, people are always breeding for higher yields, et cetera. But if you want an agent like BTH or ISR to work, you actually have to breed and select for varieties that actually, and genotypes that work. And if you do select for that, you could probably even get higher responses than what we're getting here, because that genotype effect is like anything else. You can start to breed and select for that. So we had talked to some of the people, for instance, at CIMID about this. There's some interest, but of course then when the breeders hear about this, that they think it's bad enough that you have to introduce the disease to do some breeding, but then if you have to introduce an activator and the disease to do some breeding, they just like to do breeding where you just plant it, you measure the yield at the end. They don't like things to be overly complicated there. But yeah, I'm sure with more genes examined, we would see something, at least for the SAR. For the ISR, you have to actually look at something after infection, but we've shown, many of those genes are, just like other people, are inducible by infection, more highly induced. But which ones are actually different between a responsive and non-responsive cultivar, that's not been studied so well.
Another question here? No? Well, if not, we thank you, Paul, for this nice